So I'd like to welcome everyone to our workshop on adaptive epigenomics. The theme of the workshop is to build a bridge between animal and human research. And before we get started, I'll just give you a few bits of information about the city and who organized it and details about the workshop that you need to know. So first, I'd like to say welcome to Konstanz. It's located on the Bodensee, which is bordering Switzerland and Austria and Germany. So it's 3 countries all close together. The city itself has been inhabited since the Stone Age. Then it was conquered by the Roman Empire. And then in the Middle Ages, it became a spiritual center. And currently, it's known as a university city with one of the more well-known universities in Germany. And where we are right now in Hegna is in the Kloster. It was built as a castle in the 16th century. Now it's a monastery and a hotel where they have events. So, the workshop is organized through the University of Konstanz. It is a research-oriented university. It was founded in 1966, and it's one of the elite universities in Germany. It is a member of the German Universities Excellence Initiative. So, cool. We're also organized through the Hector Fellow Academy, which is a young academy in Germany of scientists, including professors from STEM subjects, medicine, and psychology. It's a network of 22 Hector Fellows who are professors who are really well known in Germany, and 19 young scientists, and I'm one of them, and so are the other organizers. It is known for its interdisciplinary research, and so it's multidisciplinary, cross-institutional networking and exchange. There are projects between Hector Fellows so that we can cross 2 different fields and learn more about stuff. And we also host interdisciplinary workshops and conferences. It's also involved in promoting young scientists like PhDs like me. So they have PhD positions for self-developed research ideas, postdoc positions that are between 2 labs within 2 different disciplines. And then they provide you the benefit of being a membership in an excellent research network and other further training activities like MBA programs. And the 2 Hector Fellows that are involved in the organization of this workshop are Professor Dr. Thomas Elbert, who's in the psychology department. He works on clinical psychology and neuropsychology, and what he does is look at adaptation and maladaptation of the mind, brain, and body in reaction to trauma in the lab and in conflict areas. So you'll hear a lot more about his research later today, and it's really interesting. And Professor Axel Meyer, who works in zoology and evolutionary biology. The focus of the lab is on the evolution of biodiversity and speciation, with a focus on looking at the genesis of genomic, developmental, and morphological diversity. And then to introduce the co-organizers of the workshop, Amber is somewhere in the back. She's a postdoc. She works in both of the labs, actually, between Professor Elbert and Professor Meyer. And she's using live-bearing fish as a model for the impact of maternal stress across different generations. And Daniela, over there, is a PhD candidate. She's working with Professor Elbert, and she's investigating the epigenetics of post-traumatic stress disorder and its treatment. And finally is me. I'm a PhD candidate in Professor Meyer's lab, and I'm using developmental and genomic approaches to examine diversity in cichlid fish. So here's a quick overview of the schedule. What we have today is an introduction to what epigenomics actually is. Then Professor Elbert will tell us about his research. And following a coffee break, Professor, or not Professor, I don't know, Vanya Vukojevic will tell us about the role of epigenetics in human memory. And then we'll go into a more practical portion of the workshop and cover different methods in epigenetics and have some time for open questions. And after that, we will have dinner on a boat. We're leaving to Allensbach, and it's important that we're meeting here on time because the train leaves at 6:04, I think. So, yeah, they'll announce it later. And then the program tomorrow we can go over tomorrow. But some additional information is the Wi-Fi in the conference room only is, the network is called Sol Pyramid, the name of the room, and the password is the same thing. Restrooms are located in the coffee area downstairs. For the practical portion of the workshop, we have laptop charging stations on the back 4 tables. So you can't really, I mean, just make sure your computer is charged. And also there's a film crew, so yeah, you might be filmed a little bit, but we're mostly just filming the talks so that they can be put on the Hector Fellow website. So yeah, that's all. What? Oh, wait. Another thing, we're passing around a USB stick with all the slides for the presentation so that you can have all the information. Right.
Okay, may I quickly introduce you to our very first speaker, Tuncay Baubec. He's an SNF professor from the University of Zurich. We are very happy to have him here today, and he will give us a general introduction into epigenetics and what can we learn from it.
Thanks a lot, Daniela. I would like to start the organizers, Elbert, Daniela, and Maggie, for inviting me here for this nice conference, and also for the Hector Fellows Foundation. As you can see from my slides, in our group we're working on molecular mechanisms of epigenetics, and I will give you a short introduction about epigenetic gene regulation and epigenomics, and then I will tell you at the end of my talk what we're actually doing in our group. So our interest lies to understand how the genome is used to give rise to phenotypes. So as you all know, bacterial cells, they use one genome and they make one cell type. But on the other end of the spectrum, we have the human body, which consists of more than 200 different cell types, all with different functions and roles in the body. And this is all made up using the same genetic information. So this expansion in functionality cannot be only explained by the number of the genes, because you can see that the numbers of genes between E. coli and Homo sapiens differs by just a 6-fold approximate difference. So our main question is to understand how is the genetic information that is stored in our genome, this blueprint, used to give rise to all these different cell types in the human body. So how is this genetic information decoded? And many years ago, a famous biologist, Conrad, came up with a nice drawing which is called the epigenetic landscape, which is a classical drawing widely used by epigenetic researchers to describe epigenetic phenomena, which kind of indicates that a certain cell type has the possibility to give rise to all different final tissues of a body, and this would be the stem cells, which is pluripotent and has the possibility to express all genes in the genome. And during differentiation and development, this cell then has to take several decisions along the path of differentiation, which way it should go, and it rolls down these valleys and it ends up in a certain specified valley, which is then also the final destination of the cell. So basically, this is all regulated by gene expression, and meaning that the gene expression programs, which are specific for each of these tissues in the human body, they determine the cellular fate and the final identity of a cell. But the question is, how is this regulated exactly? How is the DNA sequence then decoded into function? So again, from Conrad, he had already at this time, which was long before genes were even known, he already depicted the underpinning of this landscape as a combination of several factors and strings that pull the landscape shape on which then the ball is rolling down the landscape. And this is very reminiscent of what we know nowadays as transcription regulatory factors, which have specific functions to bind to specific regulatory elements in the genome and then to drive gene expression. And of course, multiple combinations of various transcription factors can give rise to a very complex network of gene regulation, which then allows to shape this whole landscape on the course of differentiation. So this is basically what is also happening in the cellular differentiation in the body, where transcription factor batteries and various hierarchies of transcription factors work together to give rise to a gene expression program that is specific to a certain type. So these are more or less the basic components that regulate gene activity just based on DNA sequence information, which is also very much used in prokaryotes. But if you look at eukaryotic systems, there's more than just transcription factors to recognize DNA sequence specificities. There's a very well-established crosstalk between proteins that regulate protein stability and how gene expression is translated into proteins. There's a feedback between transcription and translation and vice versa. So there's a very complex network of cross-regulation between these three layers. And on top of that, the whole DNA has to be packaged into the nucleus, so it has to be wrapped around certain nucleosomes, which already have can affect how transcription factors bind to the genome. And on top of that, many of this, the DNA itself and the nucleosomes are decorated by various chemical modifications that have as well positive as well as negative influence on transcription regulation and so forth. So basically, this, as you can see, eukaryotes is a very, very complex way of regulating gene activity, which relies on multiple layers of regulation. And we are obviously interested in the epigenetic and chromatin layer, which makes use, as you know, of mechanisms that regulate gene activity independent of DNA sequence based on chemical modifications on DNA and the histone tails. So these chemical modifications are thought to influence how the DNA is accessed by various transcription factors that then turn genes on and off. Obviously, these modifications are relevant for development and human health, and they are heritable between cells and heritable between generations, but they're also dynamic, they can be reverted, and there's a certain interest from the pharma industry to develop drugs that target such proteins that are involved in the epigenetic regulations. So just a brief reminder, epigenetic modifications, the majority of them are on histone tails and also on the DNA. And besides the huge, the plethora of various modifications, which can be methylation, acetylation, phosphorylation, and various stages of methylation, so basically they fulfill two functions. Either they are associated with active transcription, or they are associated with compact, inaccessible, or silenced transcriptional activity of a gene. And obviously, these modifications have to be established. They have to be propagated between cells and during cell division. They can also be converted from one stage to the other, and as well, they can be also removed. And obviously, this all requires certain proteins that mediates these transitions. And they are the writers and the erasers of epigenetic marks, which are acetylases, methylases, phosphorylases, and various other factors that are involved in removing these marks. So they establish a certain epigenetic state that then can be further propagated by maintenance, which can be also, one can see this also as a very short input that establishes a certain mark, and the certain mark is inherited as a type of memory during cell divisions, for instance. And they're also very, very specialized proteins that contain protein domains that have been that have specific functionalities in identifying all of these different epigenetic modifications. So they directly recognize the chemical modifications on the DNA through specialized protein folds. And so that allows these regulatory proteins to bind to specific modifications in the genome and try to then convert the epigenetic information into biological function. And what is also important is that many of these factors that write, erase, or read epigenetic modifications are mutated in human diseases. So 1/4 of recurrent cancer mutations are found to occur in chromatin regulators. And obviously, many of these chromatin regulators that are found in these cancers are also targets for treatment, and they are currently numerous compounds developed that help to block, to refine the functions of this chromatin readers, writers, and erasers. Okay, so, but then the question is, so what can we learn from epigenetic and epigenomic studies? And this is also what we try to understand in our lab. So one thing that we like to look at is to try to understand when and where epigenetic modifications are deposited in the genome. So what is the spatiotemporal distribution? Meaning if you compare two different cell types, cell type A and cell type B, and we look at, let's say here, 4 different epigenetic modifications, how do these two cell types differ from each other in the epigenetic profile? Which also allows us, based on computational analysis, to try to understand crosstalk between certain epigenetic marks and certain functionalities of epigenetic marks that are related to transcriptional activity. But what we have done more in the recent years is try to understand the mechanistic background, how these epigenetic modifications are established by the writers, erasers of these marks. So how do these proteins know where to go in the genome at the right time, at the right place to establish their mark or to remove a certain mark. And then also, once these epigenetic modifications are established, which factors respond to them, which factors sense these modifications, read these out, and then how is this further translated into functions such as transcription and transcription factor binding. And then we're also interested how mutations that are found in human patients in many of these factors, how this affects the specificities of these proteins. How is a mutant protein compared to its wild-type counterpart? How is it binding to the genome? And how does this affect in the epigenetic setting and the readout of the epigenetic modifications? And then in the recent time, we started to make use of the knowledge about these mechanisms of epigenetic regulation and try to involve into epigenetic editing and engineering by reverse engineering many of these processes that can be identified in the cell, and trying to ask, can we make use of this knowledge to drive gene expression based on designed proteins that combine several of these domains here. And mainly we use one of our favorite modification, I guess many of you also use this as well in your studies, is DNA methylation. I guess I don't need to talk much about this, but just as a reminder, this is a chemical modification of methylcytosine, which is established on the double-stranded DNA by the de novo DNA methyltransferases DMT3A and 3B. This is maintained during replication by the maintenance methyltransferase DNMT1, and also in the recent years, several proteins have been identified that allow to actively remove DNA methylation from the DNA, which is mainly the TET oxygenases and also TDG proteins involved in DNA repair. And classically, DNA methylation is viewed as a transcription repressing epigenetic mark based on classical experiments where reporter genes have been inserted into cells either without or with DNA methylation on the promoter, and upon integration of the same DNA sequence into the same cell at the same position in the genome, the presence of DNA methylation results in stable repression. And once this is challenged by drugs that inhibit the DNA methylation maintenance, this stable repression is then reverted back into active transcription. In mammals, DNA methyltransferases and DNA methylation in general is essential for development. Knockout of the methyltransferase DNMT1 or 3A and 3B result in early lethality. And mainly because these proteins and the DNA methylation is involved in very important features such as dosage compensation, lineage commitment, and also genome defense against parasitic retroviruses. As well, as I told you, cancer and disease, DNA methylation is a great focus in cancer research. So many cancers have a hallmark. One of the major hallmarks of cancers is aberrant DNA methylation, where many promoter regions of imported genes are aberrantly methylated. They either lose methylation or gain methylation. Many factors involved in writing and reading DNA methylation, also erasing DNA methylations, are mutated in cancers and also other human diseases. And DNA methylation itself is mutagenic. A methylcytosine can very easily convert to thymine, and if this is happening in a coding region, in a regulatory region, this can give rise to somatic mutation that then affects the functionality of this region. This mark is widely used in various species. You have it in plants, you have it in various insects, and you have it in invertebrates and vertebrates. There's a slight difference in the way how DNA methylation is set between animals and plants. While in plants, DNA methylation can be set on various sequence contexts such as CG, CHG, CHH, whereas the H is everything but G. In animals, it predominantly occurs at the CpG denucleotide, although it can also be found at the CHH pattern. But beside this sequence specificities, there are also other very specific features of DNA methylation in mammals and vertebrates in general that makes them more different to other species. So in mammals and invertebrates, the entire genome is methylated. All CpGs in the genome are usually methylated, with the exception of actively transcribed promoters or CpG islands as well, and active enhancers where transcription factors are binding. Importantly, as you can see here, also the transcribed unit, the gene bodies of active genes is fully methylated. And this is also what is observed in invertebrates and plants for many species, that whereas the entire genome is unmethylated, the regions which contain methylation majority are repeats, repetitive elements, because they have to be silenced, and the bodies of transcribed genes. So again, you can find that DNA methylation here in these transcribed units of the genes seems to be an ancient feature of methylated genomes. And in the past years, many labs have contributed to obtaining a more detailed view about DNA methylation in various tissues, organs, species, and so forth. These are just a few examples from the literature. Here, this is a whole genome bisulfite mapping, a snapshot of this whole genome bisulfite mapping in medulloblastoma patients of various groups compared to the wild types. As you can see, there's many changes in DNA methylation happening. This is DNA methylation profiles during mouse development in certain stages and tissues. And as you can see, again, there are certain differences in the terms how DNA methylation is deposited along the genome. And this is again from mouse embryonic stem cells, a whole genome bisulfite mapping analysis, which will just indicate again that the entire genome is methylated. So you can look at exons, introns, repeat, introgenic sequences. They contain the majority of the CpGs within these regions are above 90% methylated, and the only exceptions are promoters, which are usually active. They lack DNA methylation. So what we try to understand is to see how are these DNA methylation patterns, which seem to be very dynamic between cell types, how are these DNA methylation patterns established by the factors that write this mark. So how do these proteins, the de novo methyltransferases especially, recognize the specific CpGs in this whole genome with millions of CpGs, and then set their marks at the right site at the right time. And for that, we focused on de novo DNA methyltransferases in mouse embryonic stem cells, and asking how these two methyltransferases, 3A and 3B, what are the genomic cues that attract them, what are the genomic cues that repel them, and how does this genomic binding of the methyltransferases then also translate into enzymatic activity. And based on the previous literature, there have been several mechanisms that's been postulated that regulate how DNA methyltransferases could be regulated. So one of them was histone modification. So there's a certain crosstalk between histone modifications and DNA methylation, which could attract or repel DNA methyltransferase activity. And this was mostly based on in vitro work and also based on structural studies. Also, nucleosomes themselves can have a positive or negative influence on the localizations of DNA methyltransferases. But and again, RNA or DNA sequence have been also suggested, and also tethering through various other proteins. But as I mentioned, most of these studies have been done on either on single locus analysis or in vitro, and it was not really clear how the DNA methyltransferases bind in the genome. And we used a very simple chromatin immunoprecipitation approach, where just for the ones who don't know how this works, usually the proteins of interest are cross-linked to chromatin affixed to the chromatin template, and then the entire chromatin is shredded into pieces. And then by using antibodies or other tools that specifically recognize the protein of interest, we can then pull out these chromatin parts out of this soup and then elute the DNA and sequence the DNA to obtain the genome-wide binding profiles of these proteins. So this is what the binding of DNA methyltransferase A and DNA methyltransferase B look like in the mouse embryonic stem cells. As you can see, they have a very broad binding profile, as you might also expect from a genome that is fully methylated. We observe that both proteins also tend to scale with the density of methylated CpGs. But what we also observed in these studies is that they are not identical. So there are regions in the genome where DMT3B seems to bind different to DMT3A and vice versa. And we got interested into these differences and tried to understand, so why do we see this difference in binding despite both proteins seem to be so similar in vitro studies and also based on the structural composition. And what we did, we started to look into the genome and try to understand what can explain this difference between DMT3A and DMT3B, and we also used linear modeling to predict the binding difference. And in the end, what we found out is that one particular histone modification allows us to explain the binding difference between DMT3A and DMT3B. Everything else, such as DNA methylation, CpG density, K4 dimethylation, K9 trimethylation, K27 methylation, were very similar between the two proteins. So they didn't really show any specificity towards any of these marks. But one particular modification, H3K36 trimethyl, was highly enriched for sites that were bound by the DMT3B enzyme, but not by the DMT3A enzyme. And if you then go back and then compare the binding of the methyltransferases with the histone methyltransferase, with the K36 trimethyl mark, you can observe that the binding that I mentioned before, which is more specific for DMT3B, correlates with peaks of the H3K36 trimethylation. As you can see here, here, and here. And what is also important to point out here, that the H3K36 trimethylation is known to locate over gene bodies, over gene units. And not over all of them, so there are a few of them which have this mark and few of them which don't have this mark, but you can observe that wherever K36 is located, we also have a DMT3B binding signal. So this H3K36 trimethylation mark is a histone modification that is established by a specialized enzyme, SETD2. And this SETD2 enzyme is known to bind to the elongating polymerase. So it travels with the polymerase along the gene. And every time it travels with the polymerase over the gene, it deposits this H3K36 mark on the transcribed gene. And therefore, it's a very nice correlation between this histone modification and transcriptional activity. So one can even use K36 trimethyl to identify transcribed genes in the genome without needing to do RNA sequencing. And this is also what we observe here. So if you then take all the genes in the genome and then bin them based on the transcriptional activity, rank them based on the transcriptional activity, and then make a meta profile of the H3K36 trimethyl distribution downstream of the transcription start site, so all the genes are oriented into this direction, we can observe that in the highly transcribed genes, the histone modification ramps up, and then it stops at some point. So it's really highly enriched at the highly transcribed genes, versus it goes down with the lower expressed genes until it's absent from all genes that are not expressed. On the other hand, just as a side notice, DNA methylation doesn't really care about the transcriptional activity. You have the gene bodies of transcribed genes here, they're all equally well methylated. And in contrast, you can see in the promoters, the highly transcribed genes are hypomethylated, versus genes which are not transcribed are, of course, more methylated. And then when we plot now the binding of the DMT3A and DMT3B enzymes along the same beans of transcribed genes, we can observe that for both 3A and 3.2, for the low expressed genes, the binding is very similar. They don't differ to each other. But if you look again at the highly transcribed genes, the DMT3B enzyme is more enriched at the high transcribed genes, whereas the 3A2 is missing at these regions. So this kind of suggested us that there's a link between this modification and the binding of the DMT3B protein. And we tried to understand, is this only specific to embryonic stem cells, or do we see this also in other cells? So what then, we took these embryonic stem cells and then differentiated them into neuroprogenitor cells in culture, and then looked specifically at genes that change transcription, genes that turn on and off. As you can see, these are two particular genes which gain activity in the neuroprogenitor cells. They express the genes, and that leads to accumulation of the H3K36 trimethyl mark over the gene bodies of these genes. And also, this is the same what we observe if we follow DMT3B binding in embryonic stem cells and neuroprogenitor cells, where we observe only in the neuroprogenitor cells an increase in binding at the gene body, suggesting that the protein follows K36 trimethyl changes. And this is also true from looking over all genes in the genome that change expression between ES and neuroprogenitor cells. There's a dependency of DMT3B binding to the relocalization of K36 trimethyl, suggesting to us that K36 could be a recruiter of 3B to active genes. However, this is all based on correlations here, so we wanted to really know, to figure out, is this really truly dependent on the modification? And to test this, we, just again to show you this a few examples on the wild type cells where we have K36 trimethylation located at the genes. The 3B localization follows K36 mark, and then this is 3A on top as a control. And then we really wanted to see, is K36 required for this localization of 3B? What we did then, we used CRISPR to knock out SETD2 and remove the K36 trimethyl mark from the genome, which, as you can see, results in loss of the signal, of the localized signal to the gene bodies and a more spread out signal throughout the genome. And then asked, how does the binding of the DMT3A and DMT3B proteins behave in these cells that lack K36 trimethyl? As you can see, for the DMT3A2, there's no difference in binding, so it still binds the same as it did before. It doesn't really care about K36 trimethyl. But of course, if you look at DMT3B binding, we observe that we lose the specific enrichment at K36 trimethyl sites, and then it starts to behave more like the DMT3A protein, suggesting to us that the histone modification here is required to recruit the protein to these regions to regulate possibly DNA methylation. So how is this regulated by, from the protein side? One important point is here that the DMT3s have a specialized domain, which is a PWWP domain. And this PWWP domain is widely conserved in proteins that have been shown to bind to histone to methylated histone tails, especially also to K36 trimethyl tails. And this is also available in the DMT3B protein. So it's a specialized cage, is an aromatic cage that can specifically recognize the K36 trimethyl. And this is also based on structural studies have been suggested to be the reason why these proteins could interact with K36 trimethyl. Also, based on in vitro studies, it has been shown that this PWWP domain, especially of the DMT3A protein, can interact with K36 trimethyl peptides in vitro. And also based on a human disease which is associated with DMT3B mutations, which is a human immunodeficiency, centromeric alterations, and facial abnormality syndrome. There are several mutants identified in the DMT3B gene. The majority of them lie in the catalytic domain and therefore abolish DNA methylation activity, and some of them also lead to an early termination of the protein. But there's one particular mutation which is a non-synonymous mutation in the PWWP domain of this protein. And we wanted then to ask, do these modifications have any influence of the binding of the protein, and is, in general, the PWWP domain required for the binding of the protein? So what we did, we engineered the DMT3B protein in the cells to carry either a mutation in one of these aromatic cage residues, which are conserved and required for the interactions with the K36 trimethyl. So we have this mutation. And we made one protein that carries this PWWP mutation, which has been identified in human patients, which is serine to proline substitution. And prolines are known to break two-dimensional structures in proteins. And therefore, we also made a control where we introduced the serine to proline substitution in a flexible domain in the region. So we have one mutation here in a beta sheet, which should disrupt the structure, and one mutation here in a flexible region which should not disrupt any structure of the protein. And we introduced these 3 variants into the cells and try to understand how do these proteins now behave, how do they bind to the genome, how does the binding relate to K76-trimethyl. And as you can see, this is again one of the plots I showed you before, where we look at only at the highly transcribed genes and the binding of the protein at the highly transcribed genes. You have in blue the wild-type protein, which scales again with the highly transcribed genes. It goes up like the K36 signal. If you bring in this substitution in the flexible domain, which is more a control, it also doesn't affect the binding of the protein, so it follows the same trend like the wild-type protein. However, if you introduce the mutation into the conserved aromatic residue, or if you introduce this ICF syndrome mutation, we disrupt the binding to K76-trimethyl and then lose the binding preference of the protein to this histone mark in the genome. So this kind of like explains to us how the protein senses the histone modification in the genome and allows the protein to go to the specialized regions in the genome to establish DNA methylation. But this doesn't show us that it's really required to regulate DNA methylation at this site, because so far we just looked at the binding of the protein and we don't have any measurement of the DNA methylation activity. So in order to test the DNA methylation activity, we made use of mouse embryonic stem cells, which are mutant for all DNMT enzymes. So they lack DMT1, 3A, and 3B, and therefore there is no DNA methylation in these cells. These cells can proliferate, they survive, they don't die, they do not differentiate, but they look very similar to standard embryonic stem cells. And we use the system to reintroduce either DMT3A or DMT3B, and then ask if we can so what would happen if we would reintroduce these proteins into these cells? Just to show you, these are we measured global DNA methylation using mass spectrometry. These are the triple knockout cells where you don't observe any DNA methylation in the cells. If we introduce the DMT3A to different clones, we get remethylation. If we introduce the DMT3B to different clones, we also get remethylation, although at a lower level. If we introduce DMT3B, which is a catalytic dead isoform of DMT3B6, which is a catalytic dead isoform of DMT3B, we also don't get any DNA methylation, and this is the wild-type comparison. So obviously we do not reach the same levels as in the wild-type because we don't have any maintenance mechanisms. So what is going on here, so these cells that don't have DNA methylation, the de novo methyltransferases bind to the genome, establish the methylation, and then once the cell divides, the methylation mark is lost because there is no maintenance. So every time the cell divides, the protein has to target the same site again to establish methylation. So meaning if we do whole genome bisulfite sequencing, we will be able to understand what are the targets of these proteins by just looking where is methylation set, because in order to observe the methylation, we require constant targeting of the protein to these regions. So one thing we looked at the beginning in these cell types, we wanted to see how are regulatory regions, how do regulatory regions behave in the system. And yes, we looked at active promoters and active enhancers. Enhancers are bound by transcription factors. As you can see here in the black line denotes the binding of the protein. So we also measured again the binding of the protein. As you can see, both DMT3A and DMT3B are equally excluded from active promoters and also from transcription factor binding sites, which also fits very well to the literature based on structural studies and based on in vitro studies and also other genome-wide studies, where it's very well established that there's some chromatin conformations at these regions which block DNA methyltransferases from accessing these sites. And it's not only the blocking, but also the methylation activity, which we indicate here in red or blue, depending from 3A or 3A2. You can see that the methylation is excluded from these sites, indicating that these active promoters and regulatory regions are protected from binding of the proteins and as well from de novo methylation by the proteins. And then we wanted to see how does the binding of the protein now reflect our initial observations related to K76 trimethylation dependencies. So we tried now to understand what is the difference between the de novo methylation that we measure now in these cells between 3A and 3B. So what we did, we ranked the the genome based on the amount of de novo methylation that we observe in these cells. And then in these ranked bins, we calculated the CpG density to see if there's a difference in any CpG density behavior. You can observe there's not a strong difference. There's some slight trend, which we can discuss later if you're interested. But both proteins behave the same. They follow a CpG density dependent trend, as also indicated by the genome-wide binding, suggesting that sites which have more CpGs, methylated CpGs, are also more likely to get methylated by the enzymes. But then when we did the same experiment to look at K66 trimethylation, we observed what we also observed from the binding behavior. The DMT3B, the de novo methylation by DMT3B follows K66 trimethylation mark, versus the de novo methylation activity of DMT3A is not following the K66 trimethylation mark. Again, fitting to our observations from the genome-wide binding studies of these proteins. But now again, just to make sure that this observations that we have here is indeed dependent on K66 trimethylation and not on CpG density, we took these cells where we reintroduced the DMT3B in the triple knockout cells, and then used CRISPR again to remove the SETD2 and to remove the K36. So we remove K36 in these cells where we have introduced DMT3B, and then remeasure DNA methylation after we remove the K36 modification. And as you can see from these plots, again, there's no difference in CG density. There's some slight difference, but in the end, the trend is the same. But if you look now at the K66 dependency, we observed that only in the SETD2 wild-type cells, we see the binding of DMT3B following K66, the K66 track, versus if cells lose K66, we lose this DNA methylation preference. Strongly suggesting that indeed the setting of this histone methyl mark is required to drive localization of the protein to this genomic site, and this is then in turn required to establish de novo methylation at these regions in the genome. Good. And this brings me already to the conclusion. I think I'm a bit too fast already, but I guess we plenty of time for questions. So what we observe from the studies is by looking into the binding of these de novo methyltransferases in the genome, is that in general there's a binding preference of the proteins to regions in the genome which have high CpG density. And I didn't talk about this very much, but we think this has something to do with the processivity of the enzyme. Once the enzyme starts to methylate the CpG and it senses a CpG nearby, the chance is much higher that it methylates the next CpG, and therefore it's the retention time at CpG-dense, methylated CpG-dense regions in the genome. It's much higher than at low CpG-dense regions, and this is also what we observe from the DNA methylation behavior. We observed that also the binding as well as the enzymatic activity is excluded from active regulatory regions, and also by position nucleosomes. So what's happening here, certain histone modifications such as the HTK4 trimethylation are known to block the binding of DNA methyltransferases, as well as transcription factors which compete against the DNA methyltransferases. So they protect these genomic regions such as promoter, CpG islands, and active enhancers against de novo methylation. And so these proteins are excluded from these sites. And that the majority of activity is regulated by the recruitments. Once where the protein is going, it's methylating. So there is no additional activation needed to activate the protein to set the methylation. In terms of DMT3B, which was a very surprising finding to us, we found that this protein likes to bind to this K36 trimethylated regions in the genome, which co-occur with actively transcribed genes. And this is mediated by a PWWP domain in the protein, which recognizes the K36 trimethyl mark. And what is also very striking here, that both proteins have actually this PWWP domain. And also DMT3A has been shown in vitro to bind K36 trimethyl. But we still don't understand what is the difference here. Why is only the 3B but not the 3A recruited to these regions? And we're currently working on this to try to figure out what the difference is. In any case, you could only observe that the 3B goes to these regions, and the activity and the recruitment of DMT3B to these regions is required to establish DNA methylation. Once we remove DMT3B, and there's also a recent paper that came out on this, once we remove DMT3B in these regions, DNA methylation in transcribed gene bodies is reduced, and this also leads to spurious transcriptions and aberrant transcription from these regions. So in the end, this study has shown that the 3B is required for establishment of DNA methylation at these regions. And in the end, I'd like to acknowledge my current team. These are my group members in Zurich, and also Dirk at the Friedrich Miescher, where I did my postdoc, where some of this work was done, and also funding. And thank you for your attention. I'm happy to take questions. Yes? No, so it's independent. So if you look at H3K76 in the triple knockout cells, so there is no DNA methylation at all, besides the few genes which are regulated by DNA methylation at the promoters and change transcription, of course, but the K76 is not changing. It stays the same, yes. So we have not, for this class of proteins, we have not seen preferential localization to transposable elements. So there's not, compared to other proteins where we really see strong binding to transposable elements, we haven't observed de novo methyltransferases at transposable elements. At least not at the ones which can be identified by genomic studies. You see them if you look by IF on satellite repeats. You see localization of DMT3A and 3B, but this you cannot resolve by genomics because these sites are so repetitive and not annotated. That's a good question in the field. So basically, the change from this mosaic type of methylation, as is in vertebrates, to vertebrates, which is a full genome methylation. So it's speculated what could be the reason why there is a difference. So one of the, I think, more appealing speculations is that a certain toll-like receptor has been developed that recognizes DNA and recognizes bacterial naked, unmethylated DNA, and as in terms of protective mechanisms, the genome started to be fully methylated to protect against this toll-like receptor recognizing the own genomic DNA, and this and the occurrence of this toll-like receptor and the occurrence of the whole genome methylation was approximately the same time. It's still not clear if this is really the direct reason, but there are structural studies that show that many of these toll-like receptors that recognize CPGs, they are blocked by DNA methylation. So they seem to be influenced by this. So this could be one reason. Otherwise, it could be also the types, how the de novo methyltransferases target the genome, because it could be that in these species which have only mosaic DNA methylation, they don't have this type of promiscuous whole genome methylation as you observe it in animals. They maybe have only a directed type of de novo methylation, such as small RNAs or specific histones that recruit, such as K76.
But the signal is...
Yes, the signal is because also these regions are obviously CPG rich, right? These are exons, these are transcribed genes with exons which have high CPG densities because of the conservation. And so the protein still senses the CPG rich regions in the genome. It loses the binding to the K76, but it still goes to the CPG rich sites. Yeah. That's a good question. So, so we tried to figure out if the PWP domain is responsible for this, because there are some differences in the PWP domain between 3A and 3B. So we even exchanged. So we put into 3A, we put the 3B PWP domain. We exchanged the domain and tried to see how this binding would behave, and it still didn't bind to K76. So we think it could be more like a protein-protein interaction that specifies the binding in vivo between these two proteins, despite having the same domain architecture.
Hello, thank you.
Yeah, that's a good point. So what kind of like to mention again that both the K76 trimethylation is highly conserved from yeast to man. You have it from the same deposition at transcribed genes is conserved, and also this gene body methylation that is regulated by 3B is also observed in lower invertebrates and also in plants. So that there's kind of like hints that there could be some type of functionality, that it could be required for something. And we haven't looked into overexpressing the 3B and see if this would do something, because what has been shown so far is that the loss of this gene body methylation of DMT3B and also loss of K36-trimethyl, mostly in yeast studies, this leads to aberrant transcription from within the gene body. So polymerase initiates at the wrong site in the genome, in the gene. Instead of initiating at the promoter, it starts initiating in the genome and then transcribes in the wrong direction. And of course, this affects the initial transcription. Plus, it makes these transcripts which can be problematic for the translation machinery and things like that. So it's not fully clear. The changes are minor, but it seems to have a function in this regard. Right, more robust.
Because genes are transcribed more.
Yes, yes. So you always have to think that the polymerase plows through the gene and then removes everything in front of it. So you need something that very quickly puts back the chromatin to make sure that there are no transcription factors binding at the wrong site, or there are no polymerases initiating at the wrong site. So this could be actually the function of these modifications, of both of them. They could work together, and they just reinforce.
If not, we have so first
Thank
of
you.
all, thank you again, Tuncay, for the very interesting opening and your super interesting research.
Thank you.